Biotechnology has a fascinating range of practical applications for improving the lives of others worldwide. One area of vast potential is drug design. The Sandler Center for Basic Research in Parasitic Disease at UC San Francisco Mission Bay specializes in drug design for a target that is largely neglected yet very harmful to populations throughout the developing world: parasites. The Sandler Center for Basic Research in Parasitic Disease described itself as a consortium of core laboratories to support development of new drugs for global parasitic diseases, largely ignored by the pharmaceutical industry. Their specific focus is to provide a drug development pipeline to fill a gap normally addressed in the industrial sector. I went to San Francisco to interview some of the researchers of one of these core laboratories, the McCarroll Laboratory. The McCarroll Laboratory studies a diverse group of parasitic protozoa and the biology of the host-parasite relationship. I interviewed 3 McCarroll Lab researchers, including Dr. Patricia Doyle-Engel, Dr. Zachary Mackey, and the laboratory's principal investigator, Dr. Jim McCarroll. I wanted to hear their perspectives on biotechnology's role in their research and what progress biotechnology could support in fighting various parasitic epidemics. I first interviewed Dr. Doyle-Engel, a native of Argentina who studies Trypanosoma cruzi, which causes Chagas disease.
Important disease, especially in tropical and subtropical regions of Latin America. It's a very serious disease. It's considered now one of the major neglected tropical diseases in the world. About 10 million people are currently infected, and it causes around 50,000 deaths per year. My main interest is in cell biology of T. cruzi, so I focus on cell-based assays and animal trials in the drug development pipeline at the Sandler Center. Over the past years, about 12 to 15 years, we've targeted mostly the major cysteine protease of T. cruzi, which is the parasitic agent of the disease. And we have developed inhibitors that block the activity of the enzyme, and they also prevent its autocatalytical processing and prevent the normal localization of the enzyme, and that divides actively within the cytoplasm of the host cell. So this is the stage of the parasite that causes most of the pathogenesis, and that's the one we are targeting. Over these years, we've had a number of effective inhibitors that work both in cell culture assays and in animal models of disease, and one of these inhibitors, K1177, which is a dipeptide, is in late preclinical trials right now, and we are hopeful that it will be one of the future chemotherapeutic agent trials.
Next, I spoke with Dr. Mackey about the parasite Trypanosoma brucei, which causes human African trypanosomiasis, or African sleeping sickness.
It's called Trypanosoma brucei. And Trypanosoma brucei is the causative agent of a disease that's called human African trypanosomiasis. We also refer to it as sleeping sickness. And it's called sleeping sickness primarily because the victims of the disease suffer from lethargic behavior, coma, and eventually die from the parasite. The biggest biotech advance for Trypanosoma brucei has been the advent of RNAi technology. So this has been a very good advance because, especially in a lot of the parasites, doing gene knockouts is not as amenable as it is in doing mammalian cells or yeast or bacteria study systems. So with the advent of RNAi technologies, this has been a great system for T. brucei because now we can study more readily the effects of gene regulations for T. brucei, and it's been very important to me as it pertains to identifying genes that are important for the parasite to survive. So with the advent of RNAi technology, I can study more genes, I can study more genes faster, and increase the throughput for identifying and validate these genes that are important for the survival of for T. brucei.
Finally, I spoke with Dr. Jim McCarroll, Principal Investigator of the McCarroll Lab.
Well, I was interested in basic biology, particularly of mammalian cells, and I was doing a postdoc, and the cells that I was looking at were macrophages, and macrophages migrate through tissue to get to sites of inflammation. So I was interested in how they migrate and how they degrade away to get through tissue. And I went to this seminar that someone gave on parasites, particularly on the schistosome parasite. And in the life cycle of the schistosome parasite, the larva goes right through skin without needing an insect bite or a wound or anything, goes right through skin. So I thought, wow, now here's a situation that's even more amazing than the macrophage, where this multicellular organism goes through skin. So that got me hooked on parasite biology. And that's, from then on, I just went from one parasite to another, and each one was even more amazing. We didn't really get into the drug design thing till about 10 years after we started with the basic biology, but what had happened was, in studying the basic biology of the parasites and how they infect the host, we started running across these enzymes that the parasites express that are unique and that are clearly important to the parasite's life cycle. So it kind of dawned on us that, well, gee, these might be good drug targets. And the thing that really drove us into trying to do something about that was really what the first challenge is, and that is for all of these parasitic diseases, because they're diseases of poor people in poor regions of the world, there's no interest in industry. Had this been cancer, heart disease, or something else, we might have done the basic discovery research, but we never would have gotten into the drug development thing. So the first challenge in making drugs for parasitic diseases, you don't have much in the way of industrial help. And then the second issue is related to that, is that by and large, because these are diseases of the tropical world and not the United States, there's not the kind of funding support that you'd find for aging, cancer, heart disease, stroke, whatever. So that's a second challenge. But and then the third challenge would be, since you're dealing with infectious organisms, you figure that sooner or later they're going to become resistant to any drug that you have. So you've got to always be thinking, either how can I overcome that drug resistance? How can I prevent that drug resistance? Or do I have the next thing coming down the line that if it does become resistant to my first drug, I have a second one ready to go? I mean, the first thing is that these are eukaryotic cells, so they tend to be more like our cells. So the first difference is that you're targeting something that kind of looks like you. And so that really designing drugs for parasites is more like designing drugs for cancer, because cancer cells derive from your own cells as well. So the issues are that you have to be careful that you don't hit a normal host cell process when you're trying to kill the parasite, because the cells are very similar. Second thing is that viruses and bacteria are much simpler in terms of their genomes, in terms of what they do. And so parasites tend to have mechanisms by which they can evade the immune response and also acquire resistance to drugs that viruses and bacteria don't have. So they're more complex, they're more highly evolved, and so you're dealing with really a more sophisticated enemy. I'd say two things. One is that these are the great global health problems of today. And so from social standpoint, you can't help but see that that having new drugs to control some of these diseases would really impact people all over the world, so that motivates you. And then secondly, the process of studying the organisms that produce these diseases and developing the drugs is very stimulating scientifically. So from an intellectual standpoint, it's also very motivating.